Actually, no, I'm not going to. All right, everybody. So there are only a few of us because lots of people are at the AP Human Geo test, but those of you guys who are at that test while we're having class, this recording is going to go through everything that you need to know, including the notes that we will have done. Okay, so before we do anything else, today what we're going to do a short write on modeling. And we'll do that first. And then we're going to talk about DNA replication. And then you guys are going to do a card sort for me. Okay. Homework tonight is the DNA replication practice, which is going to give you guys the opportunity to practice some skill A stuff. And the key is already online. So you can do it and then double check your thinking. And that will help you guys along the way to just know if you're on target with what you're doing. Okay. So today is all about replication. Now, I am hoping that all of you did the reading. There was a reading quiz online on replication and mitosis, which we'll be doing starting in our next class. But you guys should already have a good background knowledge, right? And part of this is making sure that you guys encounter this as many times as possible. So you will have done your video. We're going to take notes. You're going to do the card sort. You're going to do the DNA replication in practice. And then we'll talk about it again when we come back to class one more time. That's 5 times that we will have encountered exactly the same thing, okay? So this is what we're gonna be doing. So the first thing is that if you remember in our last class, we built a DNA molecule. I believe I have just about everybody's DNA molecule, but if you have not turned that in, please make sure that you do that because that is a 20-point assignment for you. So you need to make sure that you've turned that in. But we are going to actually take a look at, or kind of explore why modeling is necessary, okay? And I'm gonna read this to you because once you go in there, you have 10 minutes to do this, or no, 5 minutes to do this. The timer starts. So you wanna make sure you know before you go in. So this is the question. One of the major parts of the scientific method is communicating your results to the wider scientific community. Watson and Crick's research on DNA structure led them to build and present a physical model to help their colleagues to understand. And you can see it in the picture behind there. Explain why they needed to build a model to go along with their research paper. And how does a model help in communication? Now you need to write 4 to 5 sentences here. That is what I am checking for. But I want you to keep in mind that in research papers, just like in our smaller paragraphs, they are done as supported writing, like a CER. So they stated a claim. Their claim was more than 1 sentence. It was a lot of paragraphs of explaining. And then they had to present the evidence to support why they made those choices. So keep that in mind, that their scientific research is just a big CER, big paper-long CER. So how is it really helpful to have a model along with that paper? That's what the question is asking. Okay, so I'm going to be pausing the recording, and those of you guys who are in class, I'm going to ask you to go in and I'm going to ask you to write your answer. Okay, you have 5 minutes to do so. So having done that, we are going to move on to talking about replication. And this is the point where everybody needs to have their spirals out and open. You want to find a spot where you've got room on a page, because when we start talking about replication, you're going to have to start, like when we start doing our model, you're going to have to do a whole new page, right? So you might want to just put the few things that we need to put down on here because we just need to define what it is. So we're going to be talking about one of the 2 jobs that DNA has, right? DNA has 2 functions. One of them is making an exact copy of the DNA molecule, right? So DNA replication. Now this is different. This is why we use a special word for this, because to replicate means exact copying. Just to say copy doesn't mean it's going to be exactly the same. So it's crucial that we understand that we're making an exact copy. And the reason that we're making this exact copy is because we are going to be using the new DNA molecule to make a brand new cell that we're also hoping is going to be an exact copy of the parent cell. This is super important for multicellular organisms because most of the time when we make new cells, it's because a cell that we have has died, right? It's been injured or infected and it's died. And so if I lose, for instance, a skin cell, I need to make sure that the cell I put in its place is going to be a skin cell and that it's going to have all of the information it needs to function as well as the previous one did, if not better. Okay, so this is why we need DNA replication. This is also this part of it is the basis for reproduction as well. The fact that I can make exact copies means that then I can pass that on to a new organism. So this is why we have this. Now, before we actually outline the nuts and bolts of the process, we are going to watch a short video. Now, this is a very basic video. It is there just to make sure that we kind of have been introduced to the enzyme names, which you should have in the reading, but also that you have a mental picture of what this is going to look like as a process. Okay, so here we go. All right, so as you can see, this is, making a copy is not as simple as just copying. There are a whole bunch of proteins that are going to be responsible for making this stuff happen, right? Oops, hang on. There we go. They're going to be responsible for making this stuff happen. So what we're going to do now is we're going to talk about what those things are. We're going to nuts and bolts this out to make sure that you guys have a really good understanding of what's going on here. So we're going to switch to sketch notes, and I'm going to be doing it over here. So I'm going to stop my big share here. And I'm going to, for those of you guys at home, I'm going to be spotlighting here so that you guys can see. And hang on one second here. Do full screen. Yeah, I need you to spotlight here. That is really interesting. It is not doing that the way it should. I don't think I've had this happen before. Nice. Okay, let's remove the spotlight for a second. And usually it's really good about doing this. Try and spotlight you again. Spotlight for everybody. There we go. Okay, that was weird. Don't know why that happened. Okay, so you guys are going to be here, and you guys, although, yeah, focus-wise, it's going to be a problem for you guys here in class. So I'm going to switch over to my Elmo. There we go. And you guys should be able to see as well. All right. Now, in order to do this, those of you guys at home, so Kaylee, Scarlett, Zimri, Crystal, you guys might want to have some colors available because that's going to be helpful. But what we're going to do is this, I'm going to be doing this as a, as you guys have it in your notebook, okay? So actually, I'm going to switch the spotlight here again for just a second so that everybody can see what I'm doing. Okay, so this is your paper, and you may need a whole new page. This would be where the edge of your spiral is. So what we're going to do in order to make this work today is we are going to fold this in half lengthwise. Ooh, we have a ghost in the home, right? So we're folding it in half so that, right, like a flap. Everybody got that? Okay. Actually, well, actually it doesn't really, wait. Yeah, don't use that side, use the other side. Yep, that side. Okay, now, what you're gonna do with this now is you're gonna take this edge that you just folded so that it's touching the rings of your notebook, and you're gonna fold that in. And when you do that, you're gonna end up with two flaps. Right, so it looks like this. So it's gonna look like this. That means you can do this, right? And we can open it back up again. So it should be just like this. So folding it in half to begin with, and then folding that half back to the center. All right, everybody at home, give me a thumbs up if we're good. Awesome. Okay, so I'm going to go back to my iPad spotlight, and I'm going to remove the spotlight off myself here. Oh, actually, no, I'm going to put that back on for just a second because I need to give you guys a, okay, so we've got both spotlights on. So I am going to put this guy down because this is where we're going to be writing our notes right here. But the first thing that we're, and actually, wow, I'm making all kinds of spot decisions here. I'm going to remove this for the time being, but I'll come back to it. Okay, so the first thing that we need to do is we're going to need to make sure that we have a DNA molecule that we can work with on here. So this part right here is going to be, for one strand of our DNA, and this is going to be for our second strand of DNA. And we've already built the molecule, so we don't need to put all of the details in. So we're going to kind of simplify this out just a little bit. And this is how we're going to do it. So I'm going to start with my base. I'm going to be putting an A in place here. And if you can you see how I put this right on the edge here? So that it's here, but it's not on my flap. And I'm going to label this as an A. Okay, and I'm going to put my sugar and my phosphate on just as a stick because I don't really need much more than that to go through this. Okay, so here's my first nucleotide, and now I'm going to put another nucleotide in place. This is a T, and I have my second nucleotide in place. These guys are attached by a covalent bond, so I'm going to put that covalent bond in place. Okay, we're going to put 6 nucleotides in place here. I'm going to do my next one. My next one is going to be a C, and its nucleotide, and a covalent bond attaching that to the nucleotide above it. I'm going to make a second C, again with its nucleotide and a covalent bond attaching it. I'm going to make my next one another A, with a covalent bond. And my last one, I'm going to make a G, just because we haven't put one on there yet. And there's my covalent bond. Okay, so I have 6 nucleotides in this strand. And again, I need to make sure that I know which part is the top and which part is the bottom. So I'm going to label that specifically on this molecule. So top and bottom, so that I can see that. All right. So everybody at home, give me a thumbs up if you're good. Awesome. Okay, so now what we're going to do, we're going to flip this guy upside down, and we're going to put our complementary strand in there, but we're going to put it here on this piece, right? We're going to put it on this flap. So that, and I'm going to draw the first one here. So my G is going to be attached to a C, and here is my nucleotide. I want to be able to pull this apart, right? This is what I want to be able to happen here. So this is C, and you guys can go ahead and fill in your nucleotides. And when you're done with that, you again want to label the top and the bottom. When you get done, your molecule should be able to open and close. You'll notice we haven't used any of our colors yet, right? We're not using any of our colors. Our colors are going to be our working pieces, not our DNA, because you guys already know DNA, because you're experts now. Okay, everybody in here, good? Nick, you still working? Okay. Everybody at home, how are we doing? Thumbs up if you're done. All right. Nick, let me know when you're finished. All right, so I'm going to split my spotlight here because I want to, again, give you guys a 3-dimensional think about what this is going to be like. Now, my molecule that the molecule you just drew, this is 2-dimensional, right? So you have to imagine the fact that this is actually part of the double helix, and the code is on the inside. So if I'm going to make a copy of the code, because this is not my code here, remember, this is exactly the same all the way down in every molecule. I have to get to the code. So before I can do anything, I am going to have to take this molecule and I'm going to have to unwind it so that I can see the code. And then once I do that, I am going to need to be able to copy the code on the inside. And to do that, I'm going to have to open this up in here. So the way that this works is that there is an enzyme that comes in and it grabs onto the helix in a particular spot, and it basically just unwinds it in that one spot, and then it does what we call an unzipping, and it's going to pull this apart so that it creates a space where other workers can get in. Okay, that's the whole purpose. And we call this little space here, this is called a replication bubble, because it looks like a bubble when we look at it underneath the microscope. So, kind of difficult to make this happen on a 2-dimensional piece of paper, but we're going to do our very best, okay? So this guy right here, this is our molecule. And this place right here is the part that I just opened up on my model. So we're going to create a replication bubble here. Now, to do this, I need that enzyme to come in. And that enzyme is going to grab on right in the middle of this here. And we're going to draw that molecule that's going to do that. I'm picking the first of my colors. I happen to be using orange. It doesn't matter what color it is. Part of it is going to land here. We're actually going to have a complex, which means more than one, that are going to attach themselves onto the DNA. Okay, it's attaching itself onto the DNA. Now, I'm going to just kind of shade this in to make it a little bit more visible, this guy right here. So 2 of them are going to come down. They're going to grab onto this part. Now, these guys, and I'm going to turn this sideways so that I can label it. This is step number 1. In step number 1, this enzyme, whose name is helicase. Helicase means the ASE means enzyme. Helix comes from helix. So this is a helix enzyme, and its job is to unwind and unzip the helix. So it's going to do that right at this spot. Now, it acts like a zipper, like a double zipper. So if you think about jackets where you've got a zipper pull, this has got 2 zipper pulls. One of them is going to move in one direction, unzipping, and the other one's going to move in the other direction, unzipping, to make that replication bubble bigger. Okay? So we're going to show that on the diagram by, and actually, you guys are not seeing this at home very well, are you? I'm going to, okay, there we go. Okay, so here is my helix right here. My helicase just unwound and unzipped the helix. This zipper pull is going in this direction, and this zipper pull is going in this direction. Now, as it does that, it is going to be opening the helix up to make this space right in here, which is the replication bubble. And I'm going to label that down here at the bottom. The replication bubble is just the space that's opening up in between. And this is creating space where the workers can get in to do their job. Now, the other problem with this is that when I unwind and unzip the DNA, it wants to go back into its double helix. So we're going to have a second set of workers, another color, I'm going to use green, that are going to hold on to that replication bubble, holding it open like this. They're going to be on the outside. And there is generally a team of 4 of them. I'm going to show them. They hold on to the sugar phosphate backbone here and hold that open. So I'm going to see that on this side, and I'm going to see that on this side. Okay, so these guys right here are basically holder proteins, right? They are holder proteins. They are, and I'm going to put this on this side so that we don't lose it. This is step number 2. These are called binding proteins. Hold the helix open. So binding proteins are going to hold the helix open, and that is happening on both sides of the molecule, but we're only going to label it here. So the binding proteins are holding this open. So now I have a fully open replication bubble. The next thing that I'm going to do is I'm going to put my first big worker in place. Now this worker is the major worker. It's going to build our new molecule, and it is only able to build in one direction. It can only build from the top to the bottom. okay? Only from top to bottom. So it's going to come into this replication bubble, and it is going to sit at the top of that molecule, wherever that molecule may have been opened. Okay, so here is the worker. Now, this worker that we see here has a name that tells me what it does. This guy is DNA polymerase. ASE because it is an enzyme. Polymer, DNA polymer, because that's what it's building, right? So it's basically the DNA polymer building enzyme. So we're going to say, I'm going to actually put this here. I'm going to put builder so that we remember who that is. So the DNA polymerase is now in place. Now, it needs some things to build the DNA out of. So I'm going to draw these over here kind of in miniature. These guys right here, these are my, what we call free nucleotides. These are nucleotides that come in the food that you eat. So you eat your food, your body digests the food, the carbs get used for energy, the proteins get used to make our proteins, the fats get used to make cell membranes, and the nucleotides are used in this process. So these free nucleotides are in place. So what the polymerase does is it grabs one of those free nucleotides and it grabs this portion right here on the molecule and it puts them together. And what is it going to put in place, Aurelie? Yes, it's going to put the complementary base pair in place. So this is going to be a T. And remember that it's building this in a anti-parallel orientation, which means that this molecule is basically going the other direction. Then the DNA polymerase, having fit this guy in place, is going to shift down one spot. Now, because this is a two-dimensional molecule, I can't show you that, right? We can't draw that in place. But this little arrow comes down, it grabs onto this guy, grabs a free nucleotide, and the free nucleotide it's going to put in place is an adenine. And here's its nucleotide. And it is going to put a covalent bond between that nucleotide and the one in front of it. And then it's going to move down. And it's going to put the next complementary base in place, which is a G. And it is going to put a covalent bond between those two nucleotides. And then it shifts down. And it's going to put our next base in place, which is a G. And it's going to put a covalent bond between them. And then it shifts down and it puts in another complementary base, which is T. And it's going to create a covalent bond between them. And it's going to shift down again until it gets to the next one, which is G. And it is going to put that guy in place. And this is going to continue until it gets to the end. Right? It's going to continue until there's nothing to put in place anymore. So in doing this, we now have an old strand over here. You'll notice we didn't do anything to the old strand. The old strand is still exactly the way it was. We just opened it up. And we're going to put a new strand in place over here. Over on this side, we have our old strand, which we will not do anything with. And actually, to save some time here, since it does exactly the same thing, we're just going to show the very beginning of this process. Now, remember that our builder can only move from top to bottom. which means that our next polymerase isn't going up here because that's the bottom. Instead, it's putting itself here because this is the top of this strand. And I'm just going to draw one nucleotide going in place here, which is my G. It is going to be moving in the opposite direction. It's going to be building in the opposite way. And it's going to continue until it can't do that anymore. Okay, does this make sense? Yes, at home, does this make sense? Thumbs up if it makes sense to you. Awesome. Okay, now I do want to point out to you one thing. Remember that we're building this guy over here, and replication is supposed to be exact copy. And if you look at the old strand, look at the new strand, right? It is exactly the same. And because I'm fitting them in with complementary base pairs, it is unlikely that I'm going to make a mistake. Very unlikely. All right. So that is the simplest part of this. Now, it would not be biology if there wasn't a twist, right? There is a twist here. And we're going to, so this is the basic portion that we have here. Oh, you know what we need to do? We need to put this, we need to write down what's happening here. So our DNA polymerase, what is it doing? This is step 3. In step 3, DNA polymerase fits a complementary free nucleotide into each base in the old strand. Okay, now remember that what we're doing here is we're looking at a teeny tiny portion of this molecule. And what we need to do right now is we need to zoom out. And we're going to zoom out by going to this part of the page, okay? So we're going to take that little portion that we were looking at and we're going to move our way out so that we can see what's going on. So this is what it looks like if I zoom out. And you guys want to use about the same amount of space on your molecule that I am on mine. Okay, we're not going to put letters in here because that's just tedious and it isn't going to help us right now. But what I have here is my DNA. This is what the replication bubble looks like. It really does look like a bubble. And I'm just going to put a bunch of lines here to show where the bases would be. Okay, now I need to think about what enzymes I have in place. And I actually have 2 enzymes in place. One of them is here, right? Moving in this direction. And one of them is here, moving in this direction. These guys, this is my helicase. And it is unzipping in both directions, making that replication bubble bigger. Okay, now when that replication bubble started, it was in the middle of that replication bubble because remember, it's getting bigger all the time. So what I'm going to do is I'm going to put one of my DNA polymerase here and one of my DNA polymerase here because this is where they started. All right, so I'm going to put DNA polymerase. Now, remember that these molecules can only build from top to bottom. So I'm going to show my new strand with a different color so that we can keep track of it. And I want you to imagine what's happening. So this guy right here, it is moving in this direction because that is top to bottom. This DNA polymerase is moving in this direction because that's top to bottom. And I'm going to kind of zoom forward. This is what it's going to do. It's going to move. This guy's moving in this direction. It is going to build a new molecule until it runs out of space. So this piece is going to be a pretty long piece, and it's going to end when I run out of DNA. Same thing is happening here, is that this guy right here is moving in this direction, and it's going to put in one whole piece until it runs out of DNA. But this guy can't move in this direction. That leaves a gap. So as soon as that replication bubble gets a little bit bigger, another DNA polymerase is going to jump on. And it's going to move from top to bottom, and it's going to build a short piece until it gets to the piece that the other guy put in place, in which case it's going to pop off. And another one is going to pop in as soon as it can do that. It's going to move from top to bottom, and it's going to put in a little piece. On this side, we have the same kind of gap. So one of the DNA polymerase is going to jump in here, but it's going to be moving this direction. And it's going to build a short piece. And this guy right here, he's going to jump on again. He's going to be moving from top to bottom, and he's going to build a short piece. Okay, now I'm going to label on one side here. The big piece is called the leading strand. But the short pieces, we didn't even know that these things existed. This was a puzzle until, remember that Watson and Crick, they figured this out in 1950s. We did not know that these little pieces existed until the end of the 1960s. The guy who came up with this idea solved this problem for everybody. His name was Okazaki. And they were named for him, as well as his getting a Nobel Prize. These are called Okazaki fragments. They are little pieces. Okay? And those little pieces are going to need to be glued together, right? They're going to need to be glued together. So we're going to use, which color? I'm going to use the green color here. They are going, so this guy, ligase, I'm just going to draw him here. He is going to glue the pieces together. All right, so this is actually step number 4. And everything that I just explained to you, we're going to put in 2 sentences, right? They're 2 very loaded sentences. The first one is the leading strand, right? We know what that is. That's this big piece, is built in one continuous piece. While small Okazaki fragments are built to fill the gaps and glued together by ligase. at that. Crazy diagram that you now know what it is, right? That's pretty awesome because that's a very complicated diagram there. All right. Now, that brings us to our very last step. In our very last step, we basically need to look at the molecule first. Okay, so once all of this is done, all of the building and the gluing and so forth, what I end up with is my two old pieces, right? My two old pieces, oops, sorry guys, you can't see at home. And my two new pieces. Notice that the new pieces are in the inside, right? They're on the inside. So these are my two new pieces. So I have two old pieces and two new pieces. And because we put these together in a complementary way, there are very few errors in there. But we need there to be no errors. And so the last thing that happens is that a specialty DNA polymerase starts at the top of the molecule on either side. It literally looks like a donut, right? So this is how it's specialty. It's actually DNA polymerase, but with a slightly different shape. It is going to roam down each of the molecules, and it is going to check for errors. Because if they don't fit, the bases stick out just a tiny little bit, it gets stuck there, it fixes it, and then it continues down. So we're going to call this the spell checker enzyme. Finds and corrects any errors. All right. Now, this whole process has a term that describes it called semi-conservative replication. Semi-conservative replication refers to this picture that we have here, which says that each new molecule is going to be old and new together. It is half conserved. Old, that's fully conserved. That didn't get changed at all. And then the other half is brand new. So conserved, not conserved. Half conserved replication. All right. How does it feel? We get what's going on here? Yes? Crystal, Scarlett, Zimri, Kaylee, how are we feeling? Awesome. Okay. Now, with this, we have several things that we need to do because this is actually also the part of the basis of what's going to be going on in mitosis in our next couple of lessons as well. But I want to give you guys another chance to show me that you get this. In Schoology, there is an assignment in there called replication card sort. When you open it, it is a slideshow, right? You're going to download the slideshow, and it's going to have pieces like a puzzle where you're going to copy the picture and you're going to basically put them in order, okay? And then you're going to submit that to me as an assignment, kind of like a little quiz on replication. But you can use your notes, you can use whatever you need to, you can ask if you need to. But you're going to basically you're going to touch and hold the picture, you're going to go to the place where you want to put it, you're going to touch and hold and paste it in place, and then you can move it around, okay? The homework is the practice, that is on paper practice, and again, the key is on there. But you guys have 20 minutes. You should be able to get the card sort done, okay? You should be able to get that done, but let me know if you have any difficulties. And remember that this This is our modeling. So we have a lot of model that's going to go on to our skill A stuff. All right, go to it. Let me know if you need any help.